The term root canal, it lands with a thud, doesn't it? It's practically shorthand for dental dread. But what if we peeled back that surface layer of anxiety? What if instead of focusing on the chair and the drill, we zoomed way in down to the microscopic battlefields, the intricate signaling pathways, the fascinating dance of biology happening right inside that tiny tooth structure? Because what seems like a straightforward, if unpleasant procedure is actually this incredible intersection of neuroscience, microbiology, immunology, and even materials science. It's far more than just taking the nerve out. It's about navigating a complex biological puzzle. Today we're diving deep into that puzzle. We're going to explore the fundamental science that governs what happens when a tooth gets into trouble and how we try to set things right. Forget the common narrative for a moment and let's explore the elegance hidden within. We'll zero in on 4 key dimensions. First, the very nature of dental pain. How does a tooth even feel things and why does it hurt the way it does? Second, the microscopic world within an infected root canal. Who are the invaders? How do they organize? And how does our body fight back? Third, the remarkable process of healing. How does bone actually regenerate after the fight is won? And finally, we'll look inwards. How does your unique biology, your genetics, your overall health influence whether that tooth ultimately heals successfully? So, let's start with pain. It's not just a simple ouch. The pain from a tooth is telling a story, a dynamic narrative reflecting what's going wrong inside. Think about the different textures of dental pain, that sharp electric zing from cold versus a deep, relentless throbbing, or the truly bizarre sensation of pain seemingly radiating from a tooth that technically should be dead. Each of these is a clue. Consider the shift. Early sensitivity might give you that quick, sharp jolt. Think biting into ice cream. But if things progress, if the pulp inside becomes irreversibly inflamed, that often morphs into a persistent throbbing ache. Why the change? It's down to a fascinating molecular cascade. As the tissue inside the tooth becomes damaged and inflamed, stressed cells and responding immune cells unleash a veritable cocktail of biochemical signals. Cytokines, prostaglandins, neuropeptides, what you might call an inflammatory soup. This soup seems to trigger a shift in which nerve fibers are shouting the loudest. That initial sharp pain, likely carried by fast-acting A-delta fibers, your rapid alert system. But the later throbbing pain, that's more closely tied to the activation of slower C fibers, the ones signaling a deeper, more persistent problem. It's not just the signals themselves. This inflammatory environment can physically change tiny channels on the nerve endings, things like TRPV1 and sodium channels, making them hyperexcitable, lowering their threshold for firing off pain signals. It's like the system turns up its own sensitivity. And what about referred pain? That neurological quirk where a problem in one tooth makes your ear or jaw ache. This points to a fundamental wiring principle in our nervous system. Convergence. Sensory nerves from different parts of your face and mouth all feed into shared processing centers in the brain stem, specifically the trigeminal nucleus. Imagine multiple phone lines converging on a single operator. When a strong pain signal comes in from one line, the problem tooth, the brain, receiving the signal at that convergence point, sometimes struggles to pinpoint the exact origin. It's like the pain map for the face gets a bit blurry because the wiring is so densely interconnected. Inflammation might even sensitize neighboring pathways, making the misinterpretation more likely. Now the paradox, pain from a dead tooth. If the nerve inside the pulp is necrotic, where's the signal coming from? Here, the battleground shifts. The pain originates not inside the tooth anymore, but in the tissue surrounding the very tip of the root, the periapical tissues. Bacterial waste products like lipopolysaccharides, LPS, leaking out from the dead pulp directly irritate nerve endings in the surrounding ligament and bone. But even more powerfully, they trigger a full-blown inflammatory response outside the tooth. Your immune system sends in the cavalry, neutrophils, macrophages, which in fighting the infection release their own potent inflammatory soup, TNF-alpha, interleukins, NGF. This soup directly activates and sensitizes the pain receptors in those surrounding tissues. It's the body's defense effort causing collateral pain damage. And here's a critical point. Persistent, strong pain signals from this area can actually cause changes higher up in the central nervous system, leading to central sensitization. The spinal cord and brain become hypersensitive, potentially making pain chronic or more widespread, even after the initial source is addressed. It's like the whole system gets put on high alert. Okay, let's zoom in on the culprits themselves, the bacteria. When a breach occurs, a crack, a deep cavity, bacteria from your mouth seize the opportunity. They invade the pulp space. As they multiply and the pulp tissue dies, the environment inside the root canal changes dramatically. Oxygen levels plummet. This creates a perfect haven for anaerobic bacteria, microbes that thrive without oxygen. Crucially, these bacteria don't just float around as individuals. They form complex, highly organized communities called biofilms. Think of plaque on your teeth, but intricately established inside the root canals. It's like a microscopic city, complete with a protective slime layer and extracellular matrix, acting like a fortress wall. This biofilm structure is key. It offers incredible protection. The hard-tooth structure shields it from the outside, and the slimy matrix protects the bacteria within from immune cells and disinfectants. It's this fortified city that root canal treatment aims to dismantle. The tiny files aren't just shaping the canal, they're physically disrupting this structure. The irrigation solutions like sodium hypochlorite aren't just rinsing, they're chemical warfare, dissolving the protective matrix and killing the inhabitants. It's a mechanical and chemical siege on a microscopic fortress. But these biofilms are tenacious. The root canal system isn't a simple smooth pipe. It's often a complex network of main canals, side branches, and tiny fins, perfect hideouts where bacteria can survive even thorough cleaning. This is often why infections persist or recur, and here's where things get really exciting looking towards the future, the concept of quorum sensing sabotage. Bacteria in biofilms don't act randomly. They communicate using chemical signals. It's called quorum sensing. When the population reaches a critical density, a quorum, the concentration of these signal molecules triggers coordinated group behaviors, building thicker walls, releasing toxins, activating defenses against antibiotics. They talk, they listen, they act together. Quorum sensing sabotage, or QSS, is the idea of disrupting this communication. Imagine jamming their signals with molecules that block their receptors so they never get the message to build the fortress, or sending false signals, tricking them into dispersing prematurely or activating self-destructive processes. Instead of just trying to kill them with harsh chemicals, we could potentially outsmart them by disrupting their social network, making them vulnerable. It's targeting their organization, not just the individual soldiers. A much more sophisticated strategy. So let's say the treatment is successful. The biofilm city is cleared. What does healing look like? Clinically, the tooth feels fine, works normally. Radiographically, on an X-ray, we look for that dark spot at the root tip, the sign of bone loss from the infection, to gradually fill in. How? Once the bacterial irritation is gone, the body's repair crews move in. Specialized bone-building cells, osteoblasts, migrate to the site. Think of them as microscopic construction workers. They start laying down new bone matrix to replace what was lost. Initially, this new bone is laid down quickly, somewhat haphazardly. It's called woven bone, like a quick patch job. But over time it remodels and matures into stronger, more organized trabecular bone, restoring the normal architecture. But can we measure the quality of this healing beyond just, is the dark spot getting smaller? This is where a fascinating mathematical concept enters the picture, fractal analysis. Healthy trabecular bone has this incredibly complex, porous, interconnected structure that repeats its complexity at different scales. It exhibits fractal properties. We can actually quantify this structural complexity using a measure called the fractal dimension, FD. A higher FD means a more complex, intricate structure. The hypothesis? As the periapical bone heals, its fractal dimension should change. The initial, less organized woven bone would have a lower FD. As it matures into complex, healthy trabecular bone, the FD should increase. By analyzing the texture on radiographs, especially detailed 3D CBCT scans, and calculating the FD, we might get a quantitative biomarker of not just if bone is filling in, but how well it's maturing, how complex and healthy its structure is becoming. It's like measuring the return of structural integrity, the mathematical beauty of the healed bone. Now, while root canals have high success rates, they aren't foolproof. Why do some fail? Sometimes it's technical. Maybe the cleaning wasn't complete due to complex anatomy. Hidden canals, extreme curves, leaving bacterial survivors in those hideouts. Or perhaps the final filling or crown on top wasn't perfectly sealed, allowing new bacteria to leak back in. That seal is critical. But beyond the procedure itself, your individual biology plays a massive role. This is where host factors and genetics come in. We're learning that subtle variations in our genes can significantly impact how our bodies handle inflammation and repair. For instance, certain small genetic variations called polymorphisms in the gene for an inflammatory messenger called interleukin-1 beta, IL-1V, have been linked to a higher risk of root canal failure. The idea is that these variations might cause a more aggressive or prolonged inflammatory response around the root tip, hindering healing. Similar links are being found for variations in genes related to other inflammatory signals like TNF-alpha and genes controlling bone remodeling like RANKL. This opens the door to a future of potentially personalized endodontics. Imagine chairside genetic tests helping assess a patient's inflammatory profile or healing potential before treatment even starts, allowing us to tailor the approach. And it's not just genes, your overall health matters immensely. Diabetes, for example, is known to impair healing. Reduced immune function and compromised blood flow, often seen in diabetics, mean the periapical tissues might not repair as efficiently. Smoking is another major negative factor. Nicotine constricts blood vessels, starving healing tissues of oxygen and nutrients, and it dampens the immune response. Smokers consistently show lower success rates. Even age can play a part, as regenerative capacity generally slows down over time. Ultimately, successful healing depends on a delicate balance, resolving inflammation effectively without causing excessive tissue breakdown. If someone has genetic variations leading to overactive tissue-degrading enzymes like MMPs, their own immune response might inadvertently damage the surrounding structures. Conversely, variations promoting growth factors might enhance healing. We're even exploring epigenetics, how environmental factors switch genes on or off in the local tissues, further influencing the outcome. So, you see, a root canal isn't just drilling and filling, it's navigating a complex biological landscape. It's understanding the language of pain signals traveling along nerves, deciphering the social networks of bacterial cities, appreciating the elegant choreography of bone regeneration potentially measured by fractal dimensions, and recognizing the profound influence of our own unique genetic and systemic makeup. Thinking about it this way transforms the procedure from a dreaded intervention into a window onto incredible biological processes. And as our understanding of these fundamental areas, neuroscience, immunology, microbiology, genetics, continues to grow, it promises to further revolutionize how we approach saving teeth, making treatments even more targeted, predictable, and successful. It's a constant, dynamic interplay between our interventions and the body's astonishing capacity to fight, repair, and heal.